you can see the medium. So the rice stalk textured medium is there, no, the whitish colored one. Okay, in the base, that is called as a medium. Okay, if you are touching the medium, now you can feel a solid texture. The texture was given by agar. Okay, so along with the medium, so for a medium preparation, we can take a we can take a broth. What kind of a broth we can take now? Vegetable broth. So first we have to take pieces of vegetables, and that can be boiled with water. Then after removal of vegetables, the water, stock water, no, boiled water, vegetable boiled water, that can be used for the plant tissue culture. Along with that, we will add a little amount of agar for a solidification, and for the initiation of a cell division and for the elongation of a cell growth, we will use hormones like a cytokinin and auxins. So that entire broth, vegetable broth, agar, and growth hormones are considered as a medium. So this is the medium, the white colored one. In the bottom of bottles, we can see, no, that is a medium. Okay. So for a plant tissue culture, it's a in vitro process. Okay. In vitro process means we are culturing the plants in a glass or a glass container. It may be a tube or a conical flask or any kind of a bottles, but it must be a glass. Okay. And in what condition we are culturing the plants? Under aseptic condition. Okay, aseptic growth means there is no any hazardous thing. It's not as a contaminated area. Okay, if you are entering into a plantish culture room, na, you have to wear a mask. You have to remove your slippers. You have to cover your foot. Okay, your mouth, your head, your body parts are mostly covered except eyes. Okay, because it may make a contaminations, because we are culturing the plants. It's more or less like a newborn baby. Okay, so how we will give a attention, how we will protect the newborn baby, the same way we have to give a protection and attention to the plant tissue culture, especially for a plantlets, okay, which is grown in a plant tissue culture method. Okay, and basic principles, what are the basic principles are there? Two basic principles are there. The first one is selection of a material. Okay, the materials must be a matured one, and it must be a disease free one. So that kind of a plant materials can be used. And the second one is isolation of a explant. So which part we are going to use, that part must be a disease free part. It may be a leaf or a stem or a root. Even we can use a germplasm, or we can use a protoplast. So that selection, isolation are most important for choosing explant. So that is the first thing. Okay, so it's a 2 mark question. What are the fundamental principles for a plant tissue culture? That 2 points you have to write. Okay, and come to the next one, maintaining explant. How we have to maintain the explant? The plant explant must be physically and chemically in a controlled condition. That means it should be a disease free one. It must be a free from chemical hazardous. Okay, so that kind of physically chemically controlled condition we have to maintain the explant. Okay, because for a sterilization process, we can surface sterilize the explant, because we should not damage the plant cells. Once the plant cells are dying, it won't produce the entire plant. So physical as well as chemical, we have to protect the explants. And in a previous points itself, we were discussed what is explant. Explants are nothing. They are the part of a plant that can be used for the regeneration of an entire plant. It may be a tissue or any parts from the plant. Okay, that's called as a explant.